We are starting the second session on recent findings on cell atlases, and Sharmila and Shyam are going to continue to chair this session. Shyam is running a little late, but Sharmila will start the session. I think it's important for us to get back on time and not make people online wait for us. Yeah.
So we have with us our speaker of this first speaker of this session, Jinmiao Chen. She's a principal investigator of Singapore Immunology Network, A-STAR.
Okay, good afternoon, everyone. Welcome back from lunch. Yeah, my name is Jinmiao. Thanks to the organizer for inviting me. Today, I would like to share our preliminary results on a project called Cell-to-Virus. In this project, we explore virus versus host interactions at the single-cell resolutions and atlas scale. So as we have learned from COVID pandemic, the virus evolved rapidly. Therefore, our technology also needs to evolve as rapidly as much so that we can combat the viruses. So for example, scientists have used a whole genome sequencing to identify the virus genome sequence. And then based on that, scientists have developed the PCR or ART kits for viral infection screening. And then later, mRNA or inactivated viral vaccine has come out to help us prevent the viral infection. Among all these advanced technologies, single-cell RNA is one of them, which can capture both the viral and the host cell transcriptome simultaneously at the single-cell resolutions. But currently, most of the existing studies have looked into the host cell transcriptomes and then built a human cell atlas based on the whole cell transcriptomes. Here, I would like to zoom in into the viral transcript and see how these two interact with each other. And there is some studies that have used single-cell RNA-seq to look into the virus and host cell interactions. For example, one study published by Ido Amik's lab, they developed an algorithm called viral track and used these algorithms to detect the viral transcripts from the individual cells that were acquired from the COVID patient samples, and they used these viral track methods to map the host and viral infection interactions and identify key signatures of severe COVID. And in my lab, we would like to do these host and virus interaction at the largest scale. In my lab, we have developed this Disco Atlas, wherein we collect the raw FASTQ files from 13,000 samples, which contains 61 million cells. So in this project, we start from the raw sequence and realign the raw against the human reference genome using the same alignment tools so that we can ensure identical gene identifiers across study and reduce the technical variations. And after that, we remove the battery effect and annotated cell types and construct transcriptomic atlases from different disease and different tissues. But here, I would like to zoom in into the FASTQ files to identify the viral transcripts within individual cells. So within this project, we have a number of aims. The major aim is to explore the viral host interactions at single-cell resolution and at the atlas scale. So the first question we'd like to address is the viral tropism. What cell type is infected by different viruses? And secondly, we'd like to investigate the virus-induced transcriptome reprogramming, and such reprogramming can be cell-type specific or cell-type independent. And furthermore, we'll investigate the relationship between viral infections and the diseases such as cancer and autoimmune diseases. As we all know, that EBV can cause cancer such as NPC, nasopharyngeal cancers. EBV is also associated with autoimmune diseases such as multiple sclerosis. So we'd like to uncover common or specific mechanisms underlying the virus-driven diseases. And lastly, as we all listen a lot about the spatial omics, so here we'd like to use a spatial RNA-seq to investigate the interactions between the virus-infected cells and the bystander cells in the tissue microenvironment. So this is the bioinformatics workflow we developed, and in this workflow, we have collected the data from 13,000 samples of single-cell RNA-seq and the 670 spatial samples. And these samples span across a wide range of tissue types and the diseases. And it currently has 61 million cells comprising 461 cell types. And we map this data against the reference genome of 1,018 human viruses. So to start with, we first use cell ranges to align the raw reads against the human reference genome, and then extract the unmapped reads, and then align the unmapped reads to the virus genome. And then we perform SAM2's view and a stringent quality control using a viral check pipeline. And then we output this viral times cell unique UMI matrix and the gene times cell UMI matrix together with the cell metadata. Combining these three matrices, we would like to build a knowledge graph to see the correlation, the relationship between cell types, diseases, and infections. And by looking at both the viral transcript and the whole cell transcript, we'd like to see the cytopathic effects of the virus on the whole cell. And furthermore, we look into the immune response in the immune microenvironment. And so far, because we have 13,000 samples, it takes a lot of time to align it to the human reference genome and then the virus genome. So far, we only complete the analysis of 5,000 samples. Among these 5,000 samples, we identify 1,000 plus samples which have a virus sequence detected. And then in total, we identify about 57 different viruses in these 5,000 samples, and in total, 1.2 million cells are have a virus detected. And then these 5,000 samples are spanned across different disease tissue types such as blood, brain, bone marrow, liver, and other tissues. It also covers a wide range of disease such as COVID, liver cancer, pancreatic cancers, and then in almost all the tissue types and all the diseases, we're able to identify viruses. And in addition to the virus, we also extract the whole cell transcriptomes and integrate these data to create a tissue atlas. We have created such atlas for 27 different tissues. Here we show an example of a liver atlas and the skin atlas. When we project the virus positive cells onto this atlas, we found that the virus positive cells can be found in the hepatocytes and the macrophages, as well as other immune cell types, such as the T cells. In the skin atlas, we found our virus positive cells are mainly found in a subtype of cells called Merkel cell, which is mainly due to the association of a virus called MCV, with CMV and a Merkel cell carcinoma. And then we look into the tissue distribution of the virus. We found that EBV can be detected in a number of nasopharynx tissues, and human astrovirus in the intestine, and the liver found in HBV found in the liver, and HPV-16 found in the head and neck tissues. And let's look into the virus and disease associations and confirm numbers of such associations that has been reported in the literature. For example, we have HHV7 found in the non-Hodgkin lymphoma, HBV found in HCC. HBV is known to be a virus that can cause HBV liver cancer. And of course, we identify a dengue virus sequence in a dengue patient, and we identify a lot of ABVs in the NPC samples, and the HPV-16 is one of the common high-risk virus for head and neck cancers. And lastly, we also identify MCV in the Merkel cell carcinoma. There is a report have shown this association between this virus and this carcinoma. So that is kind of like quality control or sanity check that the workflow is trustworthy, that it identified the well-known virus and disease association. And we also look into the viral tropism to see which virus infects what cell types. We found that 50% of the virus infects the immune cells, and another 50% of the virus infects the epithelium cells, and about 1/3 of the virus infects the fibroblasts and other cell types, such as the glial cells, germ cells, and endothelium cells. And here I would like to use one study as to showcase this project. So in this case study, we look into the HPV infection in the liver. We first integrate all the data of a liver, which contains 1.4 million cells to create a liver atlas. And then we annotate the cell types and show the heat maps on the left. And then we project the HPV positive cells onto this atlas. We found that a number of HPV cells are present in the hepatocyte cluster and also in some macrophages as well as other immune cell types such as the T cells. But as we know that HBV mainly infects the hepatocytes, and why macrophage and the T cell also have HBV transcripts detected. So that is a question, and that's a question to ask whether this is due to like contamination or like embryo RNA. So we further filter by the HBV transcript UMI counts and remove the cells that shows a low UMI count of the HBV transcripts. And after the filtering, we found that HBV is actually mainly found in the hepatocytes as well as some macrophages. We also checked the HBV receptor gene SLC10A1 expression. We found that this receptor gene is highly expressed in the hepatocytes, and this receptor gene allows the HBV to enter the hepatocytes and then replicate inside the cell. As we know, the HBV doesn't infect the macrophages. Why we detect the HBV transfer in the macrophages? Our interpretation is that maybe the macrophages can like phagocyte the infected cells and take up those debris from the infected cells. That's why we can detect the HBV transcripts in the macrophages. And similar observation was also found in the Edouard mixed viral trap paper, and he showed that in the liver, and his lab also identified numbers of HBV infected cells, and these cells are mainly hepatocytes and macrophages. And now I would like to zoom into the hepatocytes, and we found that there are 4 different subsets of hepatocytes. One is the HBV negative, which is the green cells. And in addition to the negative, we identified 3 subsets of HBV positive cells. And most of the HBV positive cells are mainly found in the HCC samples. And then we look at the differential gene expressions between these 4 subsets of hepatocytes. We found that they displayed very distinct gene expression patterns across these 4 subsets. In particular, the last subset, the HBV positive hepatocyte subset 3, we see the upregulation of numbers of MSC cluster 1 genes. This means that this population of hepatocytes are very active in MSC class 1 antigen plantations. In addition to these antigen genes, we also see some genes such as SPP1, which are involved in the immune response and inflammation. So that leads us to the next analysis, which is the pathway analysis. And we found that in these 3 subsets, different pathways are upregulated in these 3 subsets. In the first subset, we found that the main changes is in the metabolic changes, while in the subset 3, the biggest changes is inflammation and immune response, while in the second subset, we see a combination of metabolism and inflammation. So, and then this leads us to the next question is that why this hepatocyte subset 3 has high inflammation and have high MSC class 1 fermentation process. And then we next look into the viral load in these 3 subsets of hepatocytes and compare their UMI count of HBV transcripts. And indeed, the hepatocyte subset 3 has the highest UMI count of HBV transcripts. And this difference is significant statistically. And the hepatocyte 1 actually has the lowest UMI count. And next, we look into the HBV gene expressions in these 3 subsets. We found that in the ORF gene, the S gene, which is the surface antigens, only one subset express this gene, which is the hepatocyte subset 1. And all 3 subsets express this core gene and the P gene, but there is a slight shift in their mapping. So that suggests that these 3 subsets of cells, the virus gene expression is slightly different, and this may contribute to the difference in the whole cell transcripts. But I would like to highlight that this difference may be due to the heterogeneity of HCC, because our study integrated data across different cohorts, different patients. So these differences may be a combination of the heterogeneity in the HCC as well as the virus heterogeneity. And in the end, we would like to share this cell-to-virus platform for data sharing, and the scientists can query this platform to search for the cell types of interest or like disease types of virus. Then the platform will show this circular plot to visualize the abundance of gene expressions of certain virus in certain cell types and the diseases. And for the remaining 5 minutes, I would like to change gear to discuss our latest work in AI and spatial omics analysis. As we know, the HCA is moving towards spatial omics, and my lab is also very interested in how we in AI development for spatial omics, because spatial omics measures the expression of mRNA and the protein while retaining the spatial context. Therefore, we need to analyze the data in a spatially aware manner, and my lab has developed a number of AI algorithms, including SEDR for imputation and batch correction, STEM for damage reduction, and GraphST for clustering and the cell type annotation or deconvolution. And more recently, we have these spatial glue methods developed for spatial multi-omics to glue across omics for analyzing such data set. And all these AI methods is based on a graph neural networks, wherein a node represents cell or molecules, the edges represents a spatial proximities between the cells and the molecules, and a highly connected subnetwork represents a cell or molecule hub. And with this slide, I would like to introduce 3 methods that have been developed in my lab. The first method is called a STEM for interpretable damage reduction. It is based on topic modeling combined with deep generative models. It outputs a biologically meaningful low dimensions associated with gene modules. With the associated gene modules, we can annotate the low dimensions for their biological meanings, so that STEM can serve as an alternative tool to damage reduction, clustering, followed by a differential gene expression analysis. Here we apply STEM to the human lung cancer data to reveal cancer-associated fibroblasts located in the tumor edge. And the second method is called GraphST. It is a graph self-supervised learning. It can produce spatial clustering, cell type deconvolution, and the batch integration. And we apply spatial GraphST to the human breast cancer data and to reveal that the T cells infiltrated into the invasive ductal carcinoma shows an upregulation of the exhaustion markers. And the third method we developed is called spatial glue, which is developed for spatial multi-omics, wherein more than one omics are measured from the same tissue slide. Spatial glue uses graph attention networks to glue across omics to reveal a finer grain tissue structures. We have test these algorithms on the brain data, thymus data to reveal a finer grain tissue structures in these 2 data sets. And lastly, I would like to thank my collaborators and my lab members for all the fantastic work they have done. Thank you for your attention.
A few questions for the speaker.
Yeah, Jay.
So 2 quick questions. So one, you showed the subset 3 relatively low in HCV or relatively low in cell number. So I was wondering, because you're compiling data set from multiple sources, yet the outcome led to a small number of cells. So were they an observation that you saw across multiple studies, or was that subset derived from a single study?
Oh, very strong echo, but I think I get what your question is. They're from different studies. We integrate about 1.4 million cells. So most likely that all these the cells, the 3 populations are coming from different studies. But for particular this subset 3, they are coming from one study. So in particular, one study that that's a very high HBV transcript in the hepatocytes.
So it would be interesting to extrapolate the clinical implications of that. But then the second question is, can you also run the same pipeline for bacteria?
Yes, we do. So that will be the next step next step. We also plan to extend this to the spatial data also. We're already seeing these bacteria in the spatial data, such as data generated from the vision technology.
I have a question. Oh, Chen, you can go. Oh, I'm here. Oh, so in the landscape of viruses that you can detect from single-cell RNA sequencing, so one expected requirement would be poly A tails in the viral RNA, right? But then there are cases like dengue that, like previously we thought that you might not be able to detect it because the mRNA shouldn't have poly A tail, but then some detected them anyway. So in your landscape now, so have you taken a look at, like the requirement or, like anything unexpected that, like some viruses might not have poly A tail, but you could detect them anyway, or anything like human endogenous viruses, could you detect their RNA from single-cell RNA sequencing data?
Yeah, so I agree with you that with single-cell RNA-seq, we can only detect those viruses with a poly A tail mRNA. But from some of them, we also detect them, although they don't have it. So we are still trying to look into what is the underlying reasons.
So it's on progress, right? So
Yeah,
it
yeah,
means
so
you don't know,
yeah.
right? Yeah, I'm curious as well. So,
No,
yeah.
it is very, very preliminary. So yeah, so where this project starts with is like we have this Disco Atlas, and with Disco we have all the raw FASTQ files sitting in our server. We also have the band files aligned to the human reference genome in our server, and then we just feel that okay, maybe we should extract like additional things out from this band file, which is the virus infection, and then we know the virus infection like associated with many diseases. So this is very, for now, is very preliminary, yeah, non-conclusive yet.
All right, thank you.
Gina, I must have seen a lot of the papers in the microbiome literature showing that there's a lot of false positives being human reads at the microbiome. For example, they were saying that you can diagnose cancer based on the microbiome.
Yes.
But it turns out there's so-called microbiome which are actually human reads. Then there's like my colleague presented she was basically saying that the human microbiome is actually not a bad microbiome and it's kind of having and so on. So one thing I remember in the slide you showed some bovine viruses in human samples, and then the significant dengue samples had SARS-CoV-2 virus infected. So which is unexpected. Wondering what the false positive rate is for this.
Yeah. So that's a very good comment. We are aware of this like false discovery, and I think, in my opinion, I think we need to use very stringent QC step to filter all those false discoveries. But there are many sources of this false discovery, so we need to be very careful to remove most of them. But in terms of virus, we do see that the expected virus in the expected tissue. So that's kind of like positive control. But we need to be very careful about the false discovery. Yeah, I agree with you. We are still in the process of that. And then for some of the viruses we don't expect to see, right? Maybe it's because of the contamination. Another one maybe is the sequence similarity. Another thing is that maybe because our data comes from all the public data, and then in some papers they use this viral vector in the experiment. So for example, cell lines or organoid, whatever. So these viruses are maybe, some of them are due to this viral vector for the gene therapy or something similar. Yeah, you very good comments. Yeah, we need to be very careful about this false discovery.
Thank you for the talk. A question from our online community. How does HPV and hepatocyte 2 and HPV plus hepatocyte 3 differ in terms of viral sequence?
Yep, so as we can see here, 2 and 3, which is the green and orange color, so the red mapping mainly they overlap quite well, but there is a slight shift in this plot. So I think this difference is subtle, but we haven't investigated like what these subtle differences means, because this is all mRNA. So we need to look into the protein, and then we need to validate it. So we are very early stage. But the most different things we found is this subset 1 have this surface antigen gene expression, which is not seen in the other two subsets. Yeah, so we believe that, the virus behave differently in different cells, in different subset of cells, and then it does like reprogram the whole cells in either like cell type specific or like cell type independent manner. So we are very keen to like zooming into the HPV biology or like liver biology, and then call for collaboration in this hall or outside of this hall.
Okay, then let's thank you.
Thank you
Very much.
Next speaker is Wenfei Jin from Southern University of Science and Technology.
Okay, so I would like to show some progress of my research, especially it's because I'm in consortium. So firstly, so we know that although we have a lot of cell atlas projects, but a lot of data are generated by various labs. So that means an active approach for cell atlas construction. So Xu Gong and in China, he proposed that we call the human ensemble cell atlas. That means collect all the scattered data in various labs and then make a unified cell atlas. Then for unified gene portrait cell type and organ. Then for those, because once you with the unified cell atlas, then you can identify the change. Then that maybe using the cell atlas to identify the disease or even for treatment. So at the first, he lab already
Wenfei, sorry to interrupt. If you could turn your microphone on. Sorry, your camera. Your video is not on.
Oh, my video.
Yeah.
So something. So. Oh, you
so the audience can see you.
Huh?
We can't see you. We can see your slides. We can't see you.
Oh, you mean
perfect.
the video? Okay.
Thank you. There you are.
So yeah, at the pilot project, collect more than 1 million cell, and you developed the unified gen table, and for unified hierarchical annotation framework, and then for the we call it for the searching the database, then we have some implication. But now we are moving. Yeah, he already have the web that everyone can access the data. Mostly he initiated a project. We have more than 10 labs and construct a unified cell atlas. So this is a web. So this means we want to establish the cell atlas of major cell organs, and also finally develop a unified cell atlas with all available cells, and also explore the medical application. So this is a big project, but here I only talk my research. So my lab is also in this we call the unified cell atlas project, and my lab will develop single cell technology and for construct cell atlas immune cell. So firstly, I would like to introduce some development my technology. Firstly, we call the single cell poly-seq. So we adapted the smart-seq and especially extended the read length and more cut. Then we can directly get more poly-A supporting reads. Then this project get much more poly-A supporting reads. Then we can see that most reads are very close to the poly A site. So that means we can identify the poly A in each cell. And although we know that a lot of genes using multiple poly A, but at the single cell level, we found that there's 2 peaks. That means each cell usually only use one of the poly A. Although it's you can say it's using multiple at the bulk level, and then we check the high variable poly A sites that this gene almost are related with cell circle. That means cell circle should associate with the poly A site usage. Then we did annotation of those high variable poly A sites. Then we identified 6 clusters, and we found that those still enrich cell circle gene, and the 6 each of the cluster enrich specific gene term. That means each gene category with a specific function, and we see that those each category they changing with the cell circle. So we think the poly A site usage is changing during that cell circle. And just we started that on one modality. So, but if you want to get more information, people want a multi-modality. So here we know that there's well-established hECA, well-established pathogenesis, and many people already developed some we call the single cell multi-omic technology for capture the chromatin accessibility and gene expression. But usually those processes are long, so we want a even simple protocol. So at that we read a paper. At that time, the paper is on the biocarb. It's called Sherry. That means different from traditional way to perform single cell hECA. It's used the TN5 segmented the DNA RNA hybrid, then using this to get the RNA for laboratory. Then we think this is simple the process. Then we integrate this strategy into our single cell multiple omics. Then we can to identify the gene expression and the chromatin accessibility. Firstly, we get the cell nuclear, then using the TN file do a segmentation, get the chromatin accessibility information, then reverse transcription and get the DNA and hybrid, then segmentation the hybrid, then get the gene expression information. Then if you are a low input, then you're using the fix and wheel. But you also can combine with the droplets, then generate more data. So this is the data. It looks okay. And we found this project perform well. And once it's we detect RNA-seq and attack-seq, both they provide information to class the cell. But the RNA-seq and attack-seq not the same information. For example, you see the RNA-seq there's one cluster, but for attack-seq there are several clusters. And then we found that even they are same cluster, if you do the motif enrichment, in fact they have different motif. That means although they are gene expression same, but the accessibility they show some differences. And also you see the pseudotype, you can see the variation over the gene attack accessibility, but the RNA are more consistent.
Now we also developed one project we call the counterpart comparator index for project concentrate on the lineages. So for example, we infer the hematosis, and we have a cancer cell to find the cell subsets in the lineage shows strong similarity with the cancer cell. Then when we project the cancer on the subpanel shows has similarity. And for example, you find the leukemia cell mapped on the HSPC, on the T lineage, and all are on the urea side. So that means once the leukemia cell located, it induces different subtype. Now we talk about the migration and differentiation of helper stem cell and progenitor cell. So helper cells have been well studied in bone marrow, but our knowledge about the migration and differentiation of helper stem and progenitor cell across tissue is limited. So now we integrate single cell data from bone marrow, peripheral blood, stemless mobilized peripheral blood, and we do an integration analysis. We identify the subset specific marker, and in further linear we see that there some cell population differences between the different tissue. Especially we can see that the HCA and the pre-B are bone marrow specific. At the beginning, the linear is mainly from the HSC from bone marrow, but some most the performer do not have much HSC. Along the linear changing, we can see that like ribosome biogenesis, cytoplastic translation, those signal changes and short time. We also can see the changing of the gene expression in bone marrow and the peripheral blood. Now we see how the migration, and then we found that bone marrow the cell type in bone marrow cells lost the migration score, and the stromal cell in bone marrow have a strong interaction with those HPC subsets. One of the intersignal that CXCL12 and CXCL4, they show quite different in different subsets. Then we check carefully that found that the bone marrow stem cell is a stronger signal sender, and then the pre-B is a stronger signal receptor. Which is reasonable, and the CXCL can promote the cell migration. And when we see the stemmers, that we found that yes, they have their specific subsets, and the bone marrow also has, but the one population they are composed by both stemmers and cells from bone marrow. So this subset may be very specific. It have population from bone marrow and stemness. So we see that it's a very important changing point, and the stemmer specific gene and the bone marrow specific, and this changing lineage from the bone marrow to the stemness. So now we have a model that shows that the cell from bone marrow can go to the peripheral blood, and most cases maybe. But well, there's only a small subset in the bone marrow can go to the stemness, and this subset ETP developed gene. Now we talk about the tumor immune microenvironment and the immunotherapy. So we know that the hot tumor, which means with a lot immune cell infiltration, those can respond to the immune therapy much better. While the cold tumor with fewer immune cells do not respond well. And we first control the single cell transcriptome atlas or cardiovascular cancer. We can those subsets, special subsets, different patients states. And we also compare the cardiovascular normal tissue and tumor tissue. In statistic, we can found that the infant cell by deconvolution analysis, and those cells are quite different. And for example, when we see the signal called PD1, LG2, PDCT1, this is a signal. Then we check those in the stellar shake. We found that those signals are co-located. That means they are really like the cell pair. Now we look at the tumor microenvironment at a specific case, and we found that in the cardiovascular cancer, the macrophage, neutrophil, DC, they will have large number of population expansion, and the gene expression between the myeloid cell shows quite differentiate. Now, we also found that the immune inhibitor score of macrophage and neutrophil is extremely high, and the GO of the macrophage and neutrophil in tumor shows strong immune inhibition property. So we further analyze the myeloid cell, found the myeloid cell subsets, and we also found the one gene SPA alpha is a cardiovascular specific tumor specific high. And when we check the deletion model, we found with deletion of the SPA alpha really decrease the tumor growth, and those with the deletion of SPA alpha have much longer survival. And then we check the spontaneous lung cancer model. They also have very few tumor in this model, and then because of tissue that show the pattern similar as a healthy tissue. Then we further compare the tumor microenvironment in the wild-type mouse and the knockdown mouse. We found especially the myeloid cell, there a lot of cell population show the change. And one of the changes we call the TM3, which is SPA alpha knockout specific subset, is shows strong anti-tumor and shows strong antigen presentation. So this is a specific population for a tumor. And also another is the SGMD6. This is subsets also show the anti-tumor immunity. So that is the anti-tumor specific subpopulation. When we check the macrophage, we see that this is that means the macrophage with the SPA knockout shows stronger phagocytosis, and they can have strong effect on the tumor growth. And surprisingly, the T cell can also have strong effects on the tumor microenvironment. So the SPA alpha don't depend on T cell. So how this SPA knockout impact on T cell? I will check it. We found that it's T cell proliferation quicker in the SPA knockout mice. So we want to know the reason, then we do the cell-cell interaction analysis from the tumor-associated macrophage. That means how interaction with the T cell, we found the potential interaction signaling, and we also found the change the macrophage T cell crosstalk, which maybe help the knockout mice have more T cell recruitment, and the likelihood interaction may be interpreted by CCL3, CCL4, and CCL8. Then we checked which cytokine may play more important in T cell recruitment. We found CCL8 really have strongest signal. And when you're using the inhibition to see how the CCL8 expression, then we found SYK, so the strongest expression inhibition. So SPAFR defense control the macrophage T cell cutoff via SYK-BTK dependent CCL separation. Lastly, we see whether this SPAFR can really benefit immune therapy. So we found the SPAFR knockout mouse with the PD-L1 antibody immunotherapy. It shows the best results with the tumor almost no growth. Okay, so finally, I thank Wenfei Jin for conducting this study, and for my collaborator. Finally, my lab is moving to Shanghai, so I have open position for co-PI, research associate, and postdoc. If you are interested, please contact with me. Bye.
Okay, over to Sharmila.
Thank you. Our next speaker is Seitaro Nomura, who's a principal investigator at the University of Tokyo.
Hi, my name is Seitaro Nomura from the University of Tokyo, Japan. It's a great honor for me to present here today. My talk is about single-cell analysis of the heart. When we see patients, we used several equipments to evaluate cardiac phenotype and provide appropriate treatments. But the most important point is to understand the genetic and the environmental factors to regulate this cardiac disease. So we performed genomic analysis and used iPS cells, animal models, and patient samples for development of precision cardiovascular medicine. In collaboration with Kaoru Ito in RIKEN, we developed polygenic risk score for atrial fibrillation using over 200,000 people genomic data. And also, by integrating these genomic data with epigenomic data, we found that these disease SNPs were mainly regulated in transcription factor ER gamma in cardiomyocytes. So for functional analysis, we used iPS-derived cardiomyocytes to show that addition of inhibitor of ER gamma leads to the perturbation of these target genes, as well as myocardial dysfunction and so on. So these genomic analysis lead to the conclusion that disruption of enhancers regulated by ER gamma lead to atrial fibrillation in human. We also performed genomic analysis of dilated cardiomyopathy, which is a main causes of heart failure, and found that these two genotypes are main causes of severe heart failure, especially the patients with LMNA mutation shows poor prognosis. So we used CRISPR technology to generate LMNA mutation knock-in mice, which shows dilated cardiomyopathy and heart failure. Also, these mice show sarcomeric abnormality in the heart. So we performed single-cell RNA sequencing and single-cell attack sequencing to show that in the LMNA mutated knock-in cardiomyocytes, transcription factor TED1 enhancers were critically disrupted. Actually, biochemical assay showed that the mutated LMNA protein could preferentially bind to the transcription factor TED1 and recruit it to the nuclear membrane. So the model shows that nuclear membrane trapping of TED1 by mutated LMNA perturbs cardiomyocyte maturation. And also, the transcriptional data of human samples confirmed that disrupted TED1 targets, specifically in LMNA mutated cardiomyocytes. We have conducted single-cell RNA sequencing of the heart from patients with heart failure and revealed that especially in patients with severe heart failure have DNA damage positive cardiomyocytes. So we have developed the method of single-cell pathological analysis to identify DNA damage positive cardiomyocytes like this, and reveal that DNA damage positive cardiomyocytes were showed the poor prognosis in heart failure patients, and several etiologies using the cardiac samples from 178 patients. This type of analysis may lead to the development of precision cardiovascular medicine. We have also performed spatial analysis. For example, this is the result of multiplex immunostaining of the heart. This is, we have developed antibody panel like this, and this is, we use this antibody panel to analyze single-cell transcriptional or proteomic features in the heart at the single-cell level. We have now identified these cell types in the heart. So this is the result of the patients with cardiac sarcoidosis. These are granuloma lesions, which is mainly consisted of inflammatory macrophages and which are surrounded by CD8 positive cytotoxic T cells. So this is summary. Several pathological stimuli lead to DNA damage in cardiomyocytes, which converted into fading cardiomyocytes, and this phenotypic change leads to also significant change in microenvironment in the heart. Single-cell and spatial transcriptomic analysis may lead to the development of cardiovascular medicine. I would like to thank all my collaborators, especially for Dr. Yukinori Okada, for integrating me with this wonderful meeting. Thank you so much.
Maybe one question for Seitaro. If there are no, thank you. Thank you very much for your very good presentation. And I hand over to my co-chair.
Yeah, last talk of the session, Miso Park from Sungkyunkwan University, who also did a stint in my lab at GIS.
I'm Miso Park and PhD candidate in Sungkyunkwan University in Korea. I'm really honored to be presentation in here. Today, I'm describe the model development process to find the program-dependent single-cell eQTL model using IDA dataset. Yes. Many eQTL studies have been applied single-cell since sequencing technology development. Because of effect of genetic variants on gene expression incompletely explain their pathogenicity, eQTL analysis using single cell is increasing. Many single cell eQTL can deliver cell type specific genetic effect that not could be obtained from bulk RNA-seq. However, many single-cell eQTL still have a limitation by pseudo-bulk resolution. So I'm going into the challenge caused by typical workflow for single-cell eQTL. First challenge is we use the pseudo-bulk expression data because of absent eQTL tool at single-cell resolution. And data can be lost about information containing in each cell since we artificially transform the pseudo-bulk expression. Second thing is inferring cell status is not feasible. Typical single-cell eQTL couldn't consider cell status because it used a pseudo-bulk expression per person. So if we use all cell, we can capture the cell status and it can be applied eQTL model by scoring. So I suggest the program-dependent single-cell eQTL model to overcome several challenges and using all cell to find the eQTL that affect the cell status. First, to get a cell status score, I propose the gene signature model. Through co-expression analysis, genes with similar functions are modularized. The module score were calculated by averaging gene expression. Each module score can be converted to cell status score after functional analysis. By applying the module score from the gene signature model, we consider the cell status to find the program-dependent single-cell eQTL. To test the model, I used the memory B cell in IDA dataset. The reason why I selected B cell is B cell had a fewer subtype than other cell type, and so it is easy to compare with pseudo-bulk expression of pseudo-bulk eQTL results. In several modules from gene signature model, blue model was confirmed to have a B cell activation functions from functional analysis. And also in our B cell subtype annotation, IgHM high and low shows similar expression pattern with the blue module score. And IgHM gene is known as a B cell activation regulator, so it has a consistent with the blue module's function. To perform the eQTL analysis at single-cell resolution, I used the mixed effect model. In this model, to obtain the interaction effect between genotype and gene module score, I made a null model without the interaction term to compare with the full model. So this is the final model structure of program-dependent eQTL considering cell status at cell resolution. And this is the one of the results using program-dependent eQTL model. All pseudo-bulk eQTL results show positive effect direction between HLA-DRB1 gene and this variant. Particularly, IgHM high group has a larger effect than lower group. In single-cell research, we separated cell into 3 groups by blue model score. And then I checked the genetic effect. Similar effect with IgHM high and low. Top 10% model score has a more positive effect than bottom 10% group. And single-cell eQTL results not divided by model score show 0.2 genetic effect, but when divided model score, genetic effect show difference by group, and we check detailed genetic effect too. Previous blue model's case was shown as a positive control with the IgHM annotation, but our eQTL model can identify eQTL that could not be checked from cell subtype annotation. The red model has a function related with cell adhesion. And MICA1 gene, known as a regulator cell adhesion molecules. The eQTL association with the MICA1 gene showed increasing genetic effect depending on red model score too. As a result, our model was able to well identify program-dependent eQTL. Program-dependent single-cell eQTL model preserved all cells. Since all cell was used, changing cell status can be captured too. And gene signature model is good to capture the cell status. Simply, it just looked like using average gene expression, but a gene list can be changed when and who did. Gene signature model solved that complexity. However, limitation is model takes a long time for learning. If we have even more cell to test, time is increasing dramatically. So this study is conducted with Hong Yi-won in Sungkyunkwan University and Humphrey Barker in Genome Institute of Singapore. And thank you very much for attending.
Thanks very much, Miso. We'll move on to the next session then. Let's thank all the speakers in this session.